substance. And we call this as the basal lamina. Okay? So yung basal surface ng cell sa epithelial tissue are actually attached to a membrane that is made up of extracellular substance. So we call this as the basal lamina. In addition to this, the basal lamina would further rest on another sheet of extracellular substance. We call this as the lamina fibroreticularis. Okay, so another layer which is lamina fibroreticularis. So dalawa yan. Since the basal lamina is thin, so it cannot be appreciated with the aid of light microscopes. But when we combine these two layers, basal lamina and lamina fibroreticularis, this would form this basement membrane, which is thick enough to be seen under a compound light microscope. So yung nakikita natin doon sa base ng cells, tapos merong layer doon, that would be the basement membrane, which is the combined layers of basal lamina and lamina fibroreticularis. Another thing, another important characteristic of epithelial tissue that we talk about, they are also assigned with special names. So you were able to encounter these two names, mesothelium and endothelium. If it is mesothelium, it is classified as simple squamous epithelium that is found lining the body cavities. Whereas if it is endothelium, same sila ng classification, it is also considered to be simple squamous, but by this time, it is found lining the surfaces of blood and lymph vessels. So nagkaiba lang sila based on the location, but still, they are both classified as simple squamous. We also talk about that epithelial tissue would have varied functions, and the function is related to the location. For example, in the slide viewing yesterday of the skin, of the epidermis of the skin particularly, since we are aware that it is lined with stratified squamous, the keratinized type, so keratinized stratified squamous epithelium. So it has several layers of cells, thus it would offer protection. Okay, so this is a good example. The lining epithelium of the skin, which is said to be keratinized stratified squamous, that's specialized for protection. In the slides again, going back to the slide of the stomach, okay, wherein it is lined with simple columnar, and basically the main role would be for absorption. Okay, and then we also use the slide of the kidney tubules, which is specialized for filtration. I presented one of the pictures yesterday of salivary gland, which is lined with stratified cuboidal. So in that case, if it is lining the glands, mainly, they would function for secretion. Okay, so these are the varied functions of epithelial tissue. You have to always relate it with the location. In naming or in classifying epithelial tissue, the two bases would be the number of cell layer and the shape of the cell. So if it is only made up of one layer, then we classify it as simple. If it is two or more, then we classify it as stratified. And then we just observe for the shape of the cells. If it is flattened, then we classify it as squamous, cube-shaped, cuboidal, and tall, narrow cells, which are the columnar cells. So these are now the pictures. First, under the number of cell layer of a simple, below is the stratified. For the cell shapes, we have here the squamous, cuboidal, and columnar cells. So let us now start in naming the types of simple epithelia. The first one would be simple squamous epithelium, which is only made up of single layer of flattened cells. So aside from that appearance, aside from the two criteria, another way on how to identify simple squamous is that you look at the shape and at the same time the position of the nuclei. So for this type, the nuclei are found in the thickest part of the cell. So if it is flattened cell, then the nuclei would be most likely located at the center or near the center. At the same time, with regards to the shape of the nuclei, it would just follow the shape of the cell. So we expect that the nuclei would appear to be flattened as well. Okay? So it consists of flattened nuclei located at the center or near the center. So those are the ways on how to identify simple squamous epithelium. We actually use the alveoli of the lungs, the slide of the lungs, to show us the simple squamous. Another example of location would be nephrons. So I mentioned that it is only made up of single layer of cells, therefore, posible yung process of diffusion to take place easily. Here are now the pictures of simple squamous. This is the one pointed in the picture below, single layer of flattened cells. In the picture above, it is seen in the surface view. Once seen from the surface, then simple squamous would exhibit polygonal outlines. So they would appear like jigsaw puzzle. Okay? So pag nakakita ka ng ganitong appearance, para siyang jigsaw puzzle, then you name it as simple squamous epithelium. Another type of simple epithelia is simple cuboidal. So it is made up of single layer of cells that are equally tall and wide, kaya tinawag na cuboidal. So again, we need to observe for the position and the shape of the nuclei for proper identification. For a simple cuboidal type of epithelium, the nuclei are rounded and are central in terms of location. These are the examples of location where simple cuboidal may be found. In the slide viewing yesterday, we used the slide of the kidney tubules to show us the simple cuboidal. But aside from that, it is also found lining the ducts of many glands. A good example would be some segments of the major salivary gland lined with simple cuboidal epithelium. Next example of location would be the surfaces of the ovaries. Okay, so these are the locations where simple cuboidal epithelium can be found. In the pictures of simple cuboidal, ito yung nakita natin yesterday, which are the tubules of the kidneys. And here, lining the major salivary gland. As you can observe, the nuclei are rounded and are at the same time located at the center. Nakakatulong din yon yung position at saka yung shape ng nuclei for proper identification. So you need to use that as part of your observations. The third type of simple epithelia is simple columnar, made up of single layer of cells that are taller than wide. Kaya columnar cells. When we look at the position and again, the shape of the nuclei are ovoidal in terms of shape and are located near the base. Another thing, when you look at the nuclei, they are arranged in single row. Same height, diba? Same level of nuclei. So that's how to identify simple columnar. An example of location is the stomach, cardiac region of the stomach. So actually, if it is simple columnar, it would line the lower gastrointestinal tract, starting stomach up to rectum. But of course, yung upper gastrointestinal tract is lined with another type of epithelium. We have also a variant of simple columnar epithelium, which is said to be ciliated. So these are the examples of location: bronchi, uterus, and oviducts. Cilia is an example of cell surface modification found at the apical surface. And we are aware already of the role of the cilia, which is specialized for transport of fluid and mucus. So cilia are also considered to be motile structures. So the ciliary movement would allow transport of fluid and mucus with trapped particles. So mainly, meron din siyang protective role, yung cilia, if the cells are ciliated. Okay? So that would be a variant of simple columnar epithelium. These are now the pictures. Here, the picture of a single layer of cells that are that consists of nuclei found at the same height and are positioned near the base at the same time ovoidal. Also in this picture, most likely yung nakita nating slide yesterday was this one. It would show us the rugae of the stomach. Okay? Hindi nga lang well-defined na naka-straight talaga. And yung isang area lang yung naka-focus, showing us the simple columnar epithelium. Okay? So somewhat like this yesterday. Okay, another picture showing us the simple squamous. Next would be the simple columnar. Ito yung inintroduce ko rin yesterday, that in both simple columnar and pseudostratified columnar, there are specialized cells in the form of goblet cells that are considered as unicellular glands. Aside from cilia, in this type of epithelium, there could be another one of presence of cell surface modification. And we call this as the microvilli. Okay? So microvilli are shorter and finer compared with cilia. So most likely in light microscopes, they are not visible. But in some locations wherein microvilli are particularly numerous, such as in the small intestines, they are seen as fine vertical lines. Therefore, we refer to this as the brush border or the striated border. So pag naka-encounter ka ng terms na brush or striated border, that would refer to microvilli. Iba rin yung role ng microvilli. These are not considered as motile structures. They would just increase the cell surface area. So that's basically the role of microvilli. Projecting from the surface of the microvilli are glycoproteins and glycolipids that would form the outer coating of the cell, which we call it as the glycocalyx. So we already named two examples of modifications found at the apical surface. First is the cilia, and second would be the microvilli. In this picture, nandito yung microvilli, fine vertical lines nga, yung appearance kaya tinawag na brush border. Since this is a cross-section of the duodenum, so ito yung apical surface on the other side that consists of the brush border. So these are the three types of simple epithelia. Let us now proceed with stratified epithelia. So the first type is stratified squamous, which is made up of several layers of cells. And sabi ko, if it is stratified epithelia, there could be combination of cell shapes. For stratified squamous, definitely the surface layer of cells are flattened cells, kaya tinawag na stratified squamous. But it may be combined with cuboidal and columnar cells as the basal cells. Okay? And these columnar and cuboidal cells that are found at the base are actively dividing. So once the cells would mature, they are pushed towards the surface and hence they become flattened cells. Kaya makikita natin sa surface are squamous cells. But still, if it is stratified squamous, we must subclassify if it is keratinized stratified squamous or non-keratinized stratified squamous. For the keratinized stratified squamous, the one that we used yesterday was the slide of the skin, epidermis of the skin, wherein the surface layer of cells are completely covered with keratin filaments. So the cells expectedly are already anucleated and they are devoid of organelles. Hindi na natin na-identify yung individual cells because they are completely covered with keratin filaments. In that given location, which is the skin, the keratinized stratified squamous epithelium is also referred to as the dry epithelium. Okay, keratinized is also referred to as the dry epithelium. In the picture below of non-keratinized stratified squamous epithelium, microscopically, when you look at the surface layer of cells, they are still nucleated. Therefore, we can identify the individual cells because there would only be few amount of keratin. For the locations, yung ginamit nating slide, vagina, other locations would include the upper gastrointestinal tract lined with non-keratinized stratified squamous, such as the oral cavity and the esophagus. So in that given locations, we also call the non-keratinized stratified squamous as the wet epithelium. Okay? So we have here the dry epithelium and the wet epithelium. For these two other types of stratified epithelia, stratified cuboidal and stratified columnar, they are just found lining some of the glands. The one that I presented, yung picture yesterday, was the major salivary gland lined with stratified cuboidal. So ano yung characteristics if it is stratified cuboidal? It is made up of 2 to 3 layers of cuboidal cells. Okay? That would line some segments of the major salivary glands. I've mentioned earlier that some segments of the major salivary glands would also be lined with simple cuboidal. Okay? So some segments, simple cuboidal, other segments lined with stratified cuboidal. The picture below would show us the stratified columnar. If it is stratified columnar, it is made up of at least 2 layers of columnar cells. And that would be the typical feature. In some locations, aside from the 2 layers of columnar cells, the basal cells are cuboidal. So it can be in combination with cuboidal cells. An example of location would be the mammary gland. Okay. Another picture showing us the stratified squamous, particularly the non-keratinized type. Okay. For the specialized epithelia, we have here the transitional epithelium, which is exclusively found in the urinary passages. So these are the examples: renal calyxes, pelvis, ureter, urinary bladder, and part of the urethra. So this transitional epithelium may resist stretching forces. So that is basically the advantage of this epithelium lining the urinary passages. For example, the transitional epithelium lining the urinary bladder would allow the organ to accommodate larger volume of urine. Because sabi ko nga yung characteristic niya may resist stretching forces. So that's the importance. This is said to be transitional because it is intermediate between stratified squamous and stratified cuboidal. So in the relaxed state of the organ, it would resemble stratified cuboidal, the one we saw yesterday in the picture. While in the distended state, it would resemble stratified squamous. So this is the picture of the ureter, and it would resemble stratified cuboidal. The dome-shaped cells are seen at the surface, and we call this as the umbrella cells. So basically, transitional epithelium, as you can observe in the picture, is classified as stratified epithelium. Okay. Another one for the specialized types, we have here the pseudostratified columnar epithelium. We only classified pseudostratified as simple. Kaya nga tinawag na false stratified. Bakit siya naging false stratified? When we look at the cells, the nuclei are scattered. They are seen at different levels. So at single glance, as if the cells have several layers, but at a closer examination, there would only be single layer because the basal surface of all the cells would lie on the same basal lamina. Similarly, with that of simple columnar, pseudostratified columnar would also have a variant. We call this as the ciliated pseudostratified columnar epithelium, which is found in the respiratory passages, such as yung ginamit nating slide, trachea, and the main bronchi. Therefore, this variant, ciliated pseudostratified columnar epithelium, is also referred to as the respiratory epithelium because of the location. Here's now the picture of trachea lined with pseudostratified columnar epithelium. These are the nuclei which are scattered, and these are the goblet cells seen also in this type of epithelium. Okay, so those are the different types of epithelial tissue. The second part of the discussion is all about the specializations or modifications found in the different surfaces. So let us first start with the specializations found at the apical surface. So we actually named already two examples. These are the cilia and the microvilli. Again, examples of specializations found at the apical surface would be number 1, cilia, and number 2, microvilli. Third example would be stereocilia. These stereocilia are basically considered to be microvilli. But why is it named as stereocilia kung microvilli yung classification? Because when you look at it in terms of appearance, in terms of length, it is as long as cilia. Sabi ko kanina, the typical characteristic of microvilli is that they are fine and shorter compared with cilia. But in the case of stereocilia, magkapareho sila ng length ng cilia. But still, the function is similar with microvilli, would increase cell surface area. Kaya we consider this as microvilli. Stereocilia are also non-motile structures. Okay? So that would be the role of the stereocilia. So for the specializations found at the apical surface, we have 3. First, cilia. Number 2, microvilli. And number 3 would be stereocilia. Next, let us proceed with the specializations found at the basal surface. Meron din kasi included dito found laterally. So unahin muna natin yung specializations found at the basal surface. The first one is the hemidesmosome, and the second one would be the basal enfoldings of plasmalemma. Again, hemidesmosomes and basal enfoldings of plasmalemma are examples of specializations found at the basal surface. So, generally speaking, pareho sila ng function. Would facilitate attachment of the basal lamina, of the basal surface of cells into the basal lamina. Diba sabi ko, naka-attach yung basal surface ng cells sa basal lamina. Pero hindi natin nakikita yung basal lamina because this is very thin. So, this basal lamina would still rest on the lamina fibroreticularis. So, yung pinagsamang layers, ito na yung magpo-form ng basement membrane. Okay? Pero yung nakadikit sa basal surface ng cell would be the basal lamina. So, these two, hemidesmosomes and basal enfoldings of plasmalemma, would facilitate attachment of the basal surface of cells into the basal lamina. Okay? A good example of location for hemidesmosomes would still be the epidermis of the skin, but particularly the stratum basale, which is found at the base. Okay? And for the basal enfoldings of plasmalemma, that would also increase absorbing capacity. An example of location would be the renal tubule. Okay? So again, for the apical surface, the examples of specializations are cilia, microvilli, and stereocilia. For the specializations found at the basal surface, we have the hemidesmosomes and basal enfoldings of plasmalemma. So finally, we have the specializations found at the lateral surfaces. In naming this, mag-start tayo sa pinaka-topmost, the one that is nearest the apical surface. So there would actually be 4 specializations found at the lateral surface. The one closest the apical surface, so number 1, would be the zonula occludens. Okay, again, the one that is nearest the apical surface, laterally speaking, we have the zonula occludens. This zonula occludens would completely encircle the cells. So the neighboring cells are tightly fused together because of the zonula occludens. Okay? Kaya rin siya tinatawag na tight junction. So it would facilitate the attachment between the neighboring cells laterally. Okay? It would completely fuse the cells together. Tight junction or zonula occludens. Next to zonula occludens, so we have zonula occludens. Next, below the zonula occludens, would be the zonula adherens. So number 2, zonula adherens. At the point where zonula adherens is found, there would be a small gap in between the neighboring cell. Pero sabi ko kanina, one of the characteristics of epithelial tissue is that it is made up of cells that are densely packed together. And why is it so? That sabi ko, sa point na nandun yung zonula adherens, there would be a small gap in between the cells. Actually, the gaps are filled with adhesion proteins. So even though meron siyang gap, it is completely filled with adhesion proteins. Kaya pag nakita natin yung cells, they are densely packed together. Okay, so that's the second one, zonula adherens. The third example of specialization found at the lateral surfaces would be number 3, desmosomes. So we have on top, zonula occludens, next, zonula adherens, and third would be the desmosomes. An example of location would be epidermis. For both zonula adherens and zonula occludens, in terms of location, they are widely developed in simple cuboidal and simple columnar epithelia. Okay, so third, desmosome. Example of location, epidermis. We can tell that at the point where desmosome is found, there would be wider gap in between the cells compared with the one at the point where the zonula adherens is located. So mas wider na yung gap ngayon sa desmosome. Sa point kung nasaan yung desmosome. But still, same thing would apply. This wide gap is filled with adhesion proteins. Examples of these adhesion proteins are the cadherins and the integrins that would fill the gaps in between the cells. Okay? So that would be the desmosomes. Finally, the one that is nearest the base, number 4, found at the lateral surface would be the gap junction, otherwise known as the communicating junction. So we have on top, zonula occludens, next, zonula adherens, third, desmosomes, fourth, gap junction, near the base. Okay? So, we can expect, kaya siya tinawag na gap junction, widest yung gap. Sa point kung saan makikita yung gap junction. Same thing would apply. The wide gap is filled with proteins. Aside from that, the gap junction consists of tiny tubes known as the connexons. So these connexons would facilitate exchange of substances such as ions and small molecules in between the neighboring cells. So nakakatulong siya. Tube siya para magkaroon ng exchange of substances between the neighboring cells found at the gap junction. Okay, so there would be 4 samples of specializations found laterally. To sum it up, the examples of specializations found at the apical surface are the cilia, microvilli, and stereocilia. Next, those that are found at the basal surface are number 1, hemidesmosomes, and number 2, basal enfoldings of plasma lemma. For the specializations found at the lateral surfaces, number 1 would be sonula occludens, 2, sonula adherens, 3, desmosomes, and number 4, gap junction. Okay, so those are the examples of the cell modifications or specializations found at the different surfaces. Okay, so that's the end of our discussion about